Yep.
Good morning, everyone. Welcome to the discussion session of today. My name is Xinping Li. I'm a postdoc in Mala Mercy's lab, and I'm joined by Edna Norman and David Deutsch. So today we're gonna discuss the two papers that I assigned to you guys for the discussion session. And the goal is not to get into great details of either of these papers, but to give you guys a general background and to introduce some of the approaches we use when we explore function of neural circuits that underlies behavior and why we take such approach. And we have received some of the questions you guys submitted through the Google form. We're gonna try to answer some of the questions during discussion, and we're gonna come back on again to answer some of the questions at the end of the discussion and then some of the live questions that came up. And if for the sake of time your questions were not addressed, or if you have further questions, please do not hesitate to reach out to us. And we will try our best to answer them or pointing you to some further references. Okay, so we're gonna talk about brains and behavior in simple organisms today. And I hope that I don't have to convince you guys that brains are interesting and important or an organ to study. And because brain pretty much defines who we are, what we feel, what we remember, and how we behave. And because it's the first brain lecture, neuroscience lecture in this summer school series, I feel obligated to show you guys some of the drawings and images made by Roman Cajal, who is considered the first modern neuroscientist. He is the person who, through this unique staining technique, had revealed that neurons are specialized cells inside of the brain and they are individual cells instead of what's being considered as brain as a continuous matter before him. And he also made this very important hypothesis, which is his law of dynamic polarization, that you see here in his drawing, that you can see the arrows. He proposed that there are information flow through the body of a neuron, and neurons communicate each other through these junctions between them. He termed synapses. So that was more than 100 years ago. Over the course of 100 years, we have learned a lot more great details about neurons and how neurons communicate each other. You probably all seen a neuron before. It has this striking morphology that we also know that neurons communicate each other with electrical signals. When a neuron is excited, it produces voltage fluctuation, which we call action potential, and it travels down the axon of the neuron, which is a neuron that's sending the signal, to the postsynaptic neuron, which is a neuron that receives the signal. And they do so by a structure called synapse, which pretty much have chemicals at the end of the ending terminals of the presynaptic neurons, and that release to the synaptic cleft, which is this gap between the two neurons, and then excite the postsynaptic neurons. So, and over the past 100 years, the goal of neuroscience, especially system neuroscience, is to figure out how neuron circuits function, which is how neurons connect to each other, that controls behaviors. And also we want to understand how genetic materials and evolution plays a role in defining the identity of specific neurons and how they connect to each other. So you may say, okay, we just need to understand, because we know that circuits function arise from how neurons talk to each other, then you will be like, we just need to figure out what are there and how they communicate to each other. And that is not such a trivial task. So naturally, the place to start is organisms with smaller brains, fewer synapses, and shorter generation time. So I'm showing you 4 different scales along the things I've talked about, from really small brains. In this case, C. elegans is a tiny worm that's no longer than 1 millimeter, that has 302 neurons, give or take plus or minus 1 across its whole body. And the synapses at the level of few thousands, to Drosophila, fruit fly, which is the organism we're gonna spend majority of today talk about. It has tens of thousands of neurons and 100 times more of synapses, and has a generation span of about 10 days, all the way to humans. Now, this number of how many neurons exactly is in the human brain has some discrepancies, but give or take is a lot more than what you will see in C. elegans, the worm, or the Drosophila, the fly. So one thing I do want to get across is that simple organisms with small brains doesn't mean that the behavior is simple or the life is simple. So take Drosophila, the fruit fly, as example. It's extremely sensory animal. It sees very well, it smells very well, and it has mechanosensory across its body. And it uses the sensory information in a moment-to-moment base to decide how it behaves. For example, during flying, Drosophila will navigate through the base, stay on track, and get to the destination while avoid any unexpected predators or obstacles. And it also uses sensory information to quickly adjust how it interacts with its conspecifics, such as aggression, fighting between Drosophilas, and then going through complex courting rituals to eventually lead to successful mating or unsuccessful mating. Okay, so it's important to study, to begin with, smaller brains that can also produce complex behavior. And the goal is to understand how neurons are connected to each other. That means that we need to be able to see the synapse. And the synapse is such tiny structures. As you can see here, this is a cross-section of several neurons smashed together, and you can see this is one single synaptic cleft here. And it is so tiny that we cannot visualize them with traditional light microscope because it just exceeds the limit of light. So we have to use this technique, and it's the only technique that allows you to achieve synaptic resolution, which is electron microscopy. And even with that, only so I should introduce the idea of connectome. So you image, if we can see every neurons inside of the brain and establish the connections between every neuron, we call that map as connectome. So now, even we have the technique and we know theoretically how to build a connectome, it's still like really complex and long process, even for the simple worm that only have 300 neurons throughout its whole body. So this is how you do it. You fixate the animal, you cut the animal across its body to different sections, and you image each individual sections, and you might not be able to image the entire cross section, so you have to divide it into like small parts of it, small tiles of it across XY dimension, and then you put the image together, and then you do the 3D reconstruction to make it back to resume the whole 3D animal, and then you trace the identity of neurons and synapses across different sections, and then you have reconstructed neurons that images from electron microscopy. So, and then after you have the identity of the neurons and where and how they connected to the other neurons, you can produce maps we now call connectome. So here is a C. elegans connectome, and each block here with number and letters represents a neuron, and each line here represents a synapses. And the first connectome was published in Worm in 1986, I believe. That was almost 30 years ago. And that was done completely by manual tracing. And over the course of the last 30 years, until last year, we only got the complete connectome of the worm, and for both sexes. And this is the only complete connectome of an organism known to us. So, as the technique evolves, we want to move on to bigger brains with more complex behaviors. and which put Drosophila as an excellent candidate because it has a lot more neurons and produce a lot more complex behaviors, but still with a manageable amount of numbers of neurons. So in this paper that we sent out for the discussion, they took similar approach. So this, what you see here is a Drosophila brain, fruit fly brain, and then you run it through electron microscopy and you image like a small section at XY dimension, and you go through the entire slice, and you do it for about 7,000 slices. And then you have the map of just the neurons of this brain. And some of you asked, why can't you do it for bigger brains? And I want you to appreciate, like, even for tiny brains, the Drosophila has, it was only possible until very recently due to the limitation of, first of all, technique, and then time and labor, and then the computing power the modern computers allows. So for these data, to collect this set of data, just the raw data, it took 18 months to complete collecting all this data. So I just want you to appreciate how long the process is. So unlike the earlier connectome that traced in C. elegans the worm, that people use manual tracing, which is that you go from slice to slice, and then you, a human tracer will have to identify whether a segment of this slice belong together with a segment from the previous slice. Machine learning and artificial intelligence algorithm has been developed to do this process automatically and only need minimum human effort to proofread the final results that comes out of the algorithm. For example, the Flywire platform uses the exact same data set in this paper. And so this is a team based in Princeton University by Sebastian Song and Malamurthy, and they have developed algorithms that automatically segments and reconstructs between slices of the electron microscopy. And so you can see that these beautiful reconstructed neurons that only require minimum human effort. So I hope that I already convinced you that connectome is such a powerful tool for several reasons. That first of all, it is the only tool for, now that you can see all the neurons inside of a whole brain of organisms, which means that you can identify new cell types just by looking at it, instead of the traditional conventional method, you're kind of fishing in the dark. And then it also allows you to see exactly where and how two neurons communicate with each other through synaptic connections. And it can allow you to identify how one neuron talks to the rest of the brain, if you like, and to uncover some of the synapses that span the area of the whole brain. And just by study how neurons talk to each other inside the brain, you can already review some of the principles of the computation inside of the brain or local circuits, and you can start to explore some of the general principles that can be generalized across species. And so here is several publications that I highlighted, which I'm not going to go into the details of the ways and the directions that they can use, already make some very useful inference of the function or the architecture of nervous system with this connectome tool. And despite that it is a very powerful tool, I don't want you to think that it is the answer to all questions, just like we have mapped the genome, the DNA information long ago, but we're still struggling to cure disease and to answer questions that's related to DNA. Connectome is more of a map, and it's more of an entry point that for you to start to answer some of the questions that are important. And some of the questions that it doesn't answer include, for example, it doesn't explain why I am different than you, despite that we have similar brains and similar neuron types in our brain. It also doesn't explain how we behave in different states. It also So because the brain is sectioned when the animal is sacrificed, obviously you can't visualize how neuron in action during behavior. And also it doesn't show the chemicals released by the neurons and also the electrical connections between the neurons. So instead of just rely on the connectome, we have to combine different methods to integrate neural circuits, such as genetic manipulation, optical recordings manipulations, electrophysiologies, and each of these methods gives you different resolution, and it has its own unique disadvantages and advantages. So through the rest of the discussion, Edna will give you a demonstration of how do we use the connectomic data and what the data looks like and what are some of the conclusions we can draw from those data. And Judy will talk about his paper and show you an example of how we combine different approaches in together with connectomes to study functions of neural circuits that controls behavior. And I will hand over to those guys.
Good morning, everyone. I'm Edna. I'm an MD PhD candidate in the second year of my PhD, and I'll be following up on Lily's talk today to show you how we actually go about using this connectome and the interface to understand more about how brains produce behavior. So neurons are interesting, as you all know, and as Lily has already described. And we know a lot about them biologically, but we have still a lot to learn. So this is an image from 20 years ago, and it's an image of a Drosophila brain. So here's a cross-section of the brain, and here highlighted are some clusters of neurons that we know are biologically interesting because they drive certain behaviors or are important for certain sensory integration. And then a while later, so this was from last year, this is a paper by Duty showing these similar neurons that are highlighted here in much greater resolution. You can actually see their cell bodies and their processes. And then here is another image by Duty, and Duty is the author of one of the papers that you read for today, and he'll be giving the talk following mine. And this is an even greater resolution. This is a light microscopy image. And we get these images by staining materials that unique groups of cells produce that other groups of cells don't, even in the brain. And here he was able to get a lot of these processes, and we're able to find these biologically relevant neurons by their anatomy, so the area that they are in the brain, so you can see that they're sort of in these parts of the brain, as well as their morphology, so the way that they look. So you can see that their processes sort of have these distinct shapes and have distinct pathways, and you can see that more clearly here. And then here's an image from this year using our connectome that Lily was beginning to describe. And this here is in the greatest resolution that we've ever had. You can see these fine processes, but more importantly, even though this is on the scale of millimeters, we can see on the scale of nanometers how these are actually connected to one another. So that means that if you look at these neurons that are 3D reconstructed here in this electron microscopy database, if you look at a cross section, so somewhere where they're physically connected over here, you can find an area where one cell, so one cell would be here, and another cell, so for example, this one here, are connected. And I'll explain in more detail how the first cell is actually passing its chemical information that Lily was also describing directly into the next cell. So we can really see at this fine resolution that they're connected, that they're talking to each other, that they're passing their material to one another, and they might go on to drive a behavior. So for example, one of these neurons might eventually connect to another one that's going to drive a chemical and electrical signal. It's a neuron that might control a muscle that might produce movement that we see in the fly and allow it to avoid a predator or get food or get into a more comfortable position to watch a lecture. So how do we actually visualize this? So this is this neuron here, but overlaid onto an image of the whole fly brain, just so you can get a glimpse of the scale. So some of you have asked about the anatomy of the fly brain. So the fly brain is a lot smaller than the human brain, but instead of grouping the areas into sort of larger brain regions. We group them into areas called neural pills, and those neural pills are just clusters of cells. Some of them we sort of know some of their functions, and we're continuing to learn about their functions, and others we have given them names and we sort of understand their morphology and where they are, but we don't really know exactly yet what they do. And we're hoping that these tools are going to help us connect this connectomic and visualization information to what they actually do biologically. So just to give you a sense, this, for example, here is the optic lobe, so it might process some visual information. The mushroom bodies are up here, they process olfactory information. We have the central complex here in this region that has a bunch of different neuropils and is important for navigation and other things. We have antennal neurons here that process sound and other things, and all these regions are interconnected, and they may do many different functions. They might be connected to one another, and we're trying to understand how the neurons in these different neuropils are talking to one another and what they're really doing at this fine resolution. So this is one interesting neuron. And this neuron is crossing the commissure, which means it's crossing the midline of the brain, so it's quite large. But many of the neurons sort of span, and you have to remember that this is an XY cross-section, and the brain exists in XYZ. So this is just in one plane, but there's sort of many back and forth. There are many neurons sort of in front of and behind this neuron, and they can be various shapes that I'll show you later. And the annotations that you see here are synaptic annotations. So they're where this neuron contacts other neurons at that synaptic resolution that we saw before. And these annotations here are overlaid on top of one another because we're zoomed out in the brain, but actually there are over 1,300 of them just on this neuron alone. So how can we actually navigate the electron microscopy interface to find these direct synaptic connections so that we can understand which neurons are talking to which neurons, and then how they might work together biologically to drive behavior? So to navigate it, we're using this interface called Flywire, developed here at the Princeton Neuroscience Institute by Sebastian Sung's lab in collaboration with Malamurthy's lab. And for example, if we want to visualize some of these interesting biological neurons in this EM data set, we're going to look for a neuron, so this one, for example, in the data set by its location anatomically, like we mentioned before, as well as its morphology, so these distinct processes and sort of where they're found in the brain. And we do that by finding something that looks like this in this EM stack. And on the left here, you can see an XY cross-section of the electron microscopy resolution of a neuron here. So for example, imagine that we slice the neuron straight across here, and we can see that here. The neuron is selected, it's in green here, and all these other circles around it are other neurons. So if we were to select those, then we would see 3D reconstructions of those neurons next to this one. And some might be this way, some might be that way, but they're all sort of together in this cross-section. And you can see that this is cellular resolution because you can see the organelles here. And on the right side of this interface, once we select the neuron that we want here on the left side, on the right side, this 3D reconstruction appears because of the segmentation that Lily was describing and all that really amazing work that was done. And we can scroll through the neuron, right? And that way we can see the XY cross-section, and then we can scroll through the Z-plane so that we can go through this entire neuron and look for synaptic connections with other neurons at each Z-plane. So sometimes as we're going through these Z-planes, we hit a place where the segmentation might be broken. So here we might be going along, right, and selecting our neuron, and we might be scrolling, and then we hit this area where we can see that this neuron is right here, and it's no longer selected. So part of this process might involve selecting it, and then here it gives it to us in a different color because the segmentation believes that it's a different neuron, but we can see that it's the same neuron and it's just continuing. And then we might merge these two segments and then get our fully reconstructed neuron that we can see up here. And once we have this fully reconstructed neuron with all of these processes, we can go on to try to find all the synaptic connections so that they can tell us which neurons this neuron talks to. How exactly do you find a synaptic connection? So there are a couple of criteria that we use. So we look for these elements called vesicles. Vesicles are pieces of membrane that contain chemicals that transmit messengers from our presynaptic neurons, so the input, to the postsynaptic neurons, so the output. And these chemical messengers might be things like acetylcholine, or neuromodulators that you may have heard of, like dopamine and serotonin. So these vesicles line up in the fly brain at something called a T-bar, and they mount here, and then they go over the synaptic cleft here into the postsynaptic cell, or the output, and that postsynaptic cell might have a postsynaptic density. Somebody asked about differences between the fly brain and the human brain, and there are many, but at synaptic resolution, some of these elements might be different. So for example, there aren't often T-bars in the human brain, and sometimes in the fly brain and other invertebrate brains, like C. elegans that Lily was mentioning, the presynaptic neuron might connect to multiple postsynaptic neurons. Also, another difference is that when you go along the neuron to find these synaptic contacts, in human brains, many of the inputs are clustered on one end of the neuron, and many of the outputs are clustered on another end, whereas in invertebrates, such as flies, they're often mixed. And the biological relevance of some of these differences are yet to be elucidated, and we're excited to be doing some of these experiments in vivo now that we've seen some of this in the electron microscopy. So I'll talk about that at the end. And so here are examples of this and what this actually looks like. So what I'm describing here in these criteria in the actual, sorry about that, in the actual electron microscopy data set. So for example, if you look here, you can see these vesicles lining up next to the synaptic cleft. Here is the T-bar that they might dock onto. Here is the cleft, and here is a postsynaptic cell and some of its postsynaptic density. So the material would go across and relay its information into the postsynaptic cell, so that the postsynaptic cell can then transmit a chemical signal to its further downstream cells. And you can see the same here, the vesicles lining up to this postsynaptic cell, and the same here, the vesicles lining up, and then here's the cleft and transmitting their information to the postsynaptic cell. So what I said that we were going to do to find all these synaptic connections is to scroll through the Z-stacks of this neuron and look for them. So I'm going to play a video of how you might do that, and hopefully this will stream. So I've selected this neuron here on the left, and again, it's reconstructed here on the right. And if you just watch, I'm scrolling through the Z-stacks, and you can see how this neuron is sort of changing in every Z-stack, and you can see it's growing here. And you'll see this zoom out. And once it zooms out, you'll see these different annotations that are all marking areas where synaptic contacts have been made on all of these fine processes of the cell. And you've seen the scope of the cell now, what it looks like when it's being zoomed out. And we can do that. So our first challenge sort of with all of this data was, as we were talking about on the first slide, to find this information at the synaptic resolution, right? So we had this information about the biological relevance for many years, and now we want to really see very directly which neurons contact with which neurons. So we're solving this problem here. And then the second problem is how we go about dealing with all of this information and really collecting it, and then how we analyze it. So we can collect it manually, so sort of what I just showed. We can also connect it automatically. So we have algorithms now that are being developed that are really great and allow us to do this on a much faster scale. And the algorithms predict the synaptic contacts by looking at these features that we've talked about before and labeling them as synapses and then recording which neurons connect to which neurons. And sometimes they do that well, but sometimes they might mistake some of, depending on the data set. So this is a second data set at a different resolution and a different type of scanning as opposed to the one that I've been showing so far. And these, sometimes they'll make errors, but overall we've had really good success rates with that. So that means that going through all of those thousands of connections, we can do that much faster and automated. And once we do that, we can, for example, visualize all the inputs and outputs of a single cell. So here's a movie showing all of the outputs of this neuron here, and you can see me panning through the brain. And you can see the morphology of what these neurons look like in 3D along the fly brain, if you kind of remember what the fly brain shape looked like here. And you can see that some of them begin to look like they're grouped into types, right? So some of them are sort of in a similar area and they have these similar shapes, and some of them don't. So based on that, we can begin to understand which types of neurons connect with this neuron and might talk to it and might go on to drive behavior or understand some sensory information or do other integrative functions. So finally, once we have that information, which neurons talk to which neurons, we could go back to what we were talking about on the first slide, right? So in the first slide, we're talking about how we have had information like this that we've been trying to get to better and better resolution, and now we have this resolution, and we can see the cross-section and see which neurons talk to which neurons. But now that we found the inputs and outputs of, say, our neuron of interest, we can actually go back to the light microscopy and to these other resolutions, and because these were taken based on the knowledge that we have of their biological relevance that we've found so far. And we can map neurons with these morphologies and this anatomy onto light microscopy images that we've had before, and we can identify them. So now we can say, oh, this is a neuron that we already saw does this. Now I can understand how this one might talk to that one. And because of the fine genetic tools that we have available in the fly, we're able to manipulate those neurons very finely and try to figure out what happens when you manipulate one neuron, what happens to the other neuron, what happens to the output of the behavior, what happens to sensory processing, and how do they connect with each other, how do they connect with the rest of the circuit. And Judy Deutsch, who is going to be talking next, is going to be describing some incredible experiments that he's done to do exactly that, to understand how different really specific neurons work together to drive really exciting behavior. So I'll hand it over to Judy with that.
Cool, thanks. So let me share my screen. We need this. Great, cool. So now I'll use some of the background that you already got in the Lily's talk and Edna's talk to give you an example for how we can use this connectome and other tools to answer specific questions in neuroscience. So I'm David Deutsch, I'm a postdoc in Mara Murthy's lab. And the work I'm working, the work I will present is the neural basis of for a persistent internal state in Drosophila females. Now, this is a simplified view of the brain where we process sensory information that could be visual one or auditory or olfactory information. And the brain needs to do a few things such as filtering the signal, removing noise, extract complex features such as faces or complex sounds, and perhaps integrate multisensory cues. Then we have decisions that we make. And when I say decision here, I refer both to discrete decisions. For example, do we want to drink coffee or tea or perhaps whiskey? And continuous decisions such as how much do we press on the brakes during driving, or how fast do we run? But both the sensory processing and the decisions, they change over time depending on the internal state of the animal. What an internal state mean? So, for example, the motivation of the animal. If the animal did not eat for some time, then it's more likely to spend time searching for food. And also, there are already works that show that it will affect how sensitive the animal is to specific odors. So it affects the sensory processing, and it also affects the decision of the animal. In this specific work, we try to understand how these internal states, or where and how are they encoded in the brain. And we use Drosophila, as you can imagine. And in the case of Drosophila, during the mating behavior, the male, here, this is the guy, is singing to the female by opening a single wing. So he is vibrating a wing, and then the female is listening to him. So this is the hearing organ here. And these decisions of the male, whether to sing or not, how loud to sing, which song to sing to the female, these are decisions that are based both on sensory information, for example, visual information about the distance from the female or the speed of the female, but also in the internal state of the male. So for example, if the male mated lately, then he is less likely to spend time in courting a female. So this is a motivational thing. And also we know that there are shorter time scales of internal state that affect how is the male choosing to sing one song mode versus another one. So here the male makes decision, uses sensory information, and also the internal state is affecting the sensory processing and the decisions. In the female side, it's more or less the same story. So she has to make decisions such as if she's going to slow down or accelerate when she hears the male, and also eventually if she will accept the courtship and copulate with this male or not. She needs to process auditory signals, but also other signals such as pheromones and visual cues. And she also, her behavior also depends on her internal state. So for example, a female that was mated lately will be unreceptive. She will move away from a singing male, and she will most likely reject this male. So in these two examples, you can see how decision, sensory processing, and internal states are connected. But we don't know that much about how is it implemented in the brain. And I want to say that when we want to link brain and activity, or how things are in the brain to the behavior, then generally there are three things that we are doing. One of them is to look at the behavior of the animal, and it is important, and we use more and more tool to make the behavioral quantification more and more precise. So we can manipulate specific cells in the brain, and this is answering one of the questions, and then see what's the effect on the behavior. So this is one component, the behavior. The other component is the brain activity. We can either manipulate specific brain areas or probe them, or we can do both. So we can, for example, ask, are these specific neurons responding to specific sounds? Are they tuned to specific sounds or to specific visual cues? So to do this, we have to probe the activity in very specific cells. And then the last component is the connectivity. And you heard a lot about this, but it is important for me to say that we use all of them, and we actually move back and forth between these three components to understand a circuit. And in this short example, I will show you how we actually use the behavioral manipulation, probing brain activity, and the connectome to answer a specific question. Right? Now, this is answering another question, and I think it's a technical thing, but it's important to understand that we have in flies, but it's also true in mice. In flies, I think it's very strong, that we have different libraries of flies. So one type of library that we have is what I call here a where library, which means specific genetic manipulations that will allow us to express specific things in specific places. So where just says, where am I going to express something? In these neurons, in these neurons, in this brain area, or in that brain area. Then we have the what library, which means what do we want to express in these neurons? Do we want to express something that will allow us to probe the activity in this specific area or in these specific cells? Or do we want to express something that will enable us to manipulate these specific cells or this specific area, right? So by combining them, so physically we take a female, we take a male, we put them together. Sometimes it's a bit more complicated, but eventually we have a fly that expresses something in a specific place. And this is generally the way that we prepare the flies for almost any kind of experiment. Right, so coming back to the question now, I want to use this type of tools to ask how the internal state in the female brain, in this case, affects her behavioral decisions, and also the sensory processing. So here is an example of how we use this where and what libraries. What we do in this experiment is that we focus on specific cells that, based on previous literature, we suspected that they might affect the internal state of the female. And to test this, we have this behavioral paradigm where we put the female in a chamber. I will show you in a second how it looks like, but it's just a courtship chamber where we put the female, and then we activate specific neurons for 5 minutes. This is this red part here where we activate the cells. So from the where library, we choose these cells. They are called PC1 cells. You might want to remember this for the next 5 minutes. So PC1 cells are the ones that we care about here. And then in the what library, we express in the PC1 cells something that will allow us to activate these cells with light. So now we just need to turn on red light for 5 minutes, and these cells are being activated, and then we stop the activation. And since we ask about the internal states, and we know that internal states are persistent, so the animal stays hungry or stays or is affected by mating for some time after she stopped eating or stopped mating, then we are asking if there is some persistent activity or also some persistent effect on the animal behavior following the activation. So what happens 10 minutes or 20 minutes or 30 minutes after we already stopped activating this cell? Do we have any persistent activity that affects the animal behavior and sensory processing? Now that's the experiment. Here is how the chamber look like. We have microphones, so we can record the male singing. And you can see here, it's hard to see, but this is the female, this is a male, and we track them offline. So we can eventually see how the animals behaved, how the female responded to male song, how do they interact. And we can do it after different manipulations. In this specific case, we can see how activating the PC1 cells for 5 minutes affects the female behavior, the female response to the male song also. Now, to actually be able to extract not only the position, but also exactly the angle of the female. So you see, this is the head and the body of the female, and this is the male direction. With this very non-homogeneous background, we used machine learning techniques, deep networks. This is a paper published by Talmo Pereira from our lab. And doing this, we could extract many parameters that describe each frame in the movie, right? So for example, in each frame, we now know what is the distance between the fly, what is the speed of the female, what is the relative angle between the female and the male, etc. We have 17 parameters, and now we want to get the behaviors from this. Now we use two techniques, and I will talk about this briefly. One approach that is generally used to characterize behavior is unsupervised classification. This is where we take all the parameters, in our case 17 of them, and we look for clusters of behaviors. So clusters of, that describes different groupings of these 17 parameters without telling the software anything about, I'm looking for this behavior or for that behavior. So this is what we did first. And we looked for behaviors that are different between the case where we activate the PC1 neurons and the controls, right? So this is an unsupervised classification. We found two behaviors. And once we found them, we said, let's focus on these behaviors that seem to be affected by PC1 activation. And then we used another approach, which is supervised classification. And Java is just a tool for supervised classification, where we train a specific network or a specific algorithm, more precisely, to find this specific behavior based on many examples that we show to the software first. And all of this is based on these parameters that we extracted. So after doing all this process, we eventually found that there are two behaviors that happen more upon PC1 activations. I didn't get into this, and you can read it in the paper, but we also varied the delay between the activation and the time we measure. So there are more details to this, but eventually we found two behaviors. One is aggressive behaviors. It's female shoving. So the female is facing the male and pushing him back. The other one is chasing behavior, where the female is chasing the male, and it happens a lot after we activate PC1 neurons, which is here, but it doesn't happen almost at all when you just look at natural courtship. Usually the male is chasing the female, not the female is chasing the male. So we have two behaviors that persisted for many minutes after we stopped activating PC1 neurons. Now, now we established that there is some link between PC1 activation and some internal state that lasts for many minutes in the female brain that causes her to change her behavior, to have this aggressive behavior and chasing the male. And I didn't show it, but it also changes the way the female is responding to the male song. So this is another effect of the internal state. But now is the time that we wanted to use the connectome, because PC1 is a group of neurons that we knew about. But maybe PC1 is actually multiple groups of neurons. And if you look more carefully, you'll find out that there are different subtypes. And indeed, this was the case, that we found that PC1 neurons, they look more or less the same. But if you look more into the details, then you'll see that there are actually 7 major subtypes within the PC1 population. And one of them, PC1 alpha, we found it to be sufficient for driving these behaviors. Now, it's important to understand we can't get this just by using the connectome, but once we found that there are different types of subtypes of PC1, we could then use genetics to then express the same thing that we expressed before, such that now we can do the same experiment, but now activating only a subset, only PC1 alpha neurons, and again, measure what's the effect. And we found that PC1 alpha is enough to now to get this shoving and chasing behaviors. There's this aggressive behavior and the shoving one. So now we focused on PC1 alpha neurons, and we asked two more questions. One, let's see what are the inputs and outputs of PC1 alpha And the other one is let's look at the neural activity in the brain upon PC1 alpha activation. So we mapped all the inputs and outputs to PC1 alpha. One major finding was that there is strong reciprocal connectivity between PC1 alpha and 3 other groups of neurons, or it's actually one other group that has 3 subtypes. And this is important because it has been shown in the past that reciprocal connectivity could explain a persistent activity, because the same neuron is an output and an input at the same time. So you get some recurrency, and this can allow persistency in the network. So we suspect that this recurrent connectivity might explain the persistent activity, perhaps together with other mechanisms. So this is an example for how we use the connectome both to narrow down to specific subsets of PC1, but also to find this recurrent connectivity that could explain the persistent effect of PC1 alpha activation. Right? So the last step, as I told you, was to now activate the PC1 alpha neurons. So we activate them when the fly is under a microscope like this, and then we can do a volumetric imaging of the brain and see what happens in the brain after we activate these PC1 alpha neurons. And this is another case where having a small brain is a big advantage, because no one can scan the whole brain of a mouse. But we can definitely scan a large chunk of the brain, of the fly's brain. So this is one hemisphere, and we scan most of one of the hemispheres, so we can systematically check what happens in all this volume upon PC1 activation. What we find is that some areas look like this. So they are active, we activate them during the PC1 alpha activation, but then it goes back to baseline. But there are some other brain areas where the activity goes up during, and then it takes many, many minutes for the activity to go back to baseline, right? So these areas in the brain seem to carry this persistent activity, and kind of luckily for us, there is a nice correspondence between where these areas are and where we found this reciprocal connectivity that I told you about before. So it points out to a very specific circuits that might carry that persistent activity. And of course, we need to work more to understand exactly how this persistency occurs within this small area. And I'll mention shortly here that there was recently another paper that came from mice where they showing the mouse an aversive stimulus, in this case a rat, causes persistent activity for over a minute in a specific area in the mouse brain. And there are actually a few commonalities between this and what we found in the fly. But I also wanted to mention that they did some simple modeling work, and this work led them to hypothesize, and we would like to actually check if it's true. It's easier in flies, that this persistency is caused by a combination of recurrent connectivity and also the role of neuropeptides that together with this recurrent connectivity allows this minutes-long persistent activity. Right? So coming back to this schematics, we now understand, or we found a specific locus for internal state control in the female fly, and we saw how it's affecting decisional, behavioral decisions, and also auditory processing in the female. Of course, there are many open questions coming from this, but at least we focused on specific areas and we suggest some mechanism. Now I want to spend 2-3 more minutes on saying one more thing, which is that optimally, we would like to put together the behavior and the imaging. As you probably noticed, we now do the behavioral experiments in one place, and then we take the fly and do the imaging in a fixed fly where we look into the brain. But imagine that we could do both things together, that we could look at the brain activity while the female is interacting with the male in this social behavior. How can we do something like this? So generally, there are two approaches for this, not just in flies. So the first one is to use freely moving animals. In the case of rodents, for maybe 7-8 years now, it's possible to put a portable microscope, a very small microscope that moves with the mice. So then you have a moving animal and you can record brain activity. It has many caveats and limits, but this is one possible approach. Definitely we cannot do it in flies. The flies are just too small, right, to hold any microscope. The other, but in this direction, people thought that maybe in flies, what we could do is use some complicated optics such that we can track the fly and image the fly using some moving mirrors and cameras and stuff like this, while the fly is hanging around and doing its social behavior. This is an interesting approach. It's very limited, very complex today, and it has some limits to it. One of them is that we are limited in the resolution, and also if the fly is moving, tilting, hidden by the other fly, in many cases, we are missing chunks of information. So maybe in the future, some of these problems will be solved. It's not easy. The other more fundamental issue is that we have zero control over the stimulus. So if we want to we cannot manipulate anything. The flies do their thing. But if we want to ask, for example, how the response of the female to the male singing is affected by her seeing him opening his wing. For example, we want to have a male that is singing without opening a wing. Now, this is not possible. So there is another solution, which is to use virtual realities. Now we have a fixed fly that is either flying or walking on some treadmill, and we have a virtual reality for the fly. It's like a computer game where the other fly can move around or whatever complex stimulus environment we want to have. So in this case, the big disadvantage is that we cannot get the full behavioral repertoire. Obviously, a female cannot attack a male when she's just walking on the ball. On the other hand, we can image at a high resolution, and we can also have full control over the stimulus. So we can introduce a courtship song while manipulating the wings, the size of the animal, and many other things. So these are two different approaches. We are going in the lab towards the second approach more in building systems like this, where the fly is walking on the ball. We image brain activity. We have auditory stimulus. This is a visual screen. We also have olfactory stimulus that you can see here. And we can also introduce flies or other stimuli on this visual screen. So this is kind of looking into the future or the near future and asking such questions. Right, I think now we can move to the Q&A part. Thanks.
Thanks, Dudi. So a question had been asked about the role of genetics in the development of the fly brains and the differences between that role in flies and in humans. So just to address that question quickly, so the development of circuits is greatly controlled by genetics, and it is also influenced by environment, as in all or many organisms. And in both flies and in humans, the development of specific circuits, meaning the wiring of the brain, the numbers of certain neurons, the connections that they make with one another, how that happens differently in different sexes of flies, how that happens in different parts of the brain. So that is controlled often by genetics. It's controlled by transcription factors, which are proteins that have elements that can bind to DNA and then up or down regulate the expression of certain genes that are unique to certain groups of cells. So that happens in both flies and in humans. And in flies, the brains are smaller and they tend to be more stereotyped than we understand them to be in humans. So there are neurons that we can manipulate, as Dudi was describing, that are those specific cell types are existing across many flies. So we can activate, for example, PC1 neurons in the first fly and in the second fly and in the third fly. And that's how we can do these very specific experiments, because these neurons with these specific morphologies and anatomy and biological components all exist across these different flies. Now, it's more challenging to locate specific neurons that are analogous among different humans. There's more plasticity in humans, and there's just so much more variability, and so just such a greater neuron number. But what is similar is that there are cell types. So there are groups of cells that have similar morphology and anatomy. And what we can do is look at the whole human brain and try to categorize it into areas, which we've already done. And we're still continuously trying to understand what the functions of those areas are. And they often have many functions and connect with one another. And within each of those sections, we can then go further down and create subgroups of cells that maybe are, in smaller areas that are closer to one another and that look like one another. And we're trying to right now figure out how far down we can sort of classify these cell types in the human brain. One approach is I saw that there was a question about connectomics in humans, and that's a super important question because like the point of everything that we're doing in terms of methods, but also we're trying to elucidate the principles behind how brains work so that we can apply them to humans and other organisms. And in terms of the methods, what Sebastian's lab is trying to do, and other labs are, they're trying to look at specific small chunks of the human brain, or not the human brain, the mouse brain, which is a higher level model organism than flies, but lower than humans in complexity. And they're trying to get an electron microscopy data set for those chunks, because as Lily had described, it's extremely intensive and time consuming to create a whole brain data set of these higher level organisms, because it's taken so long to create the connectome of just C. elegans and the fly. And also we don't know yet how much the stereotypy will exist in these higher level organisms. So some of these principles, such as the cell types, genetic regulation, so the transcription factors that I described, and many, many other principles apply across these organisms developmentally and then also in adults, as Judy was describing, in terms of how they give rise to behavior. So those are some of the similarities and some of the differences. And the other ones, we are still trying to understand what those similarities and differences are. And even if we find that there are differences between flies and humans in terms of some of those principles, that'll still be really interesting because it'll give us a greater understanding of how some of these differences have emerged and how some of this might work in humans versus flies, and just might give us more insight. So I hope that answered some of those questions. And Judy might be able to talk more about some of those differences between humans and flies.
Right, thanks. So I answered a few specific questions in the Q&As, but let me maybe finish with answering something that many of you brought many times, which is the general question about, okay, so we do all these things in flies, the flies are simple, we can maybe understand the circuit at a higher resolution comparing to rodents, definitely comparing to humans, but if it's not relevant for humans, then why do we care? It's a fair question, and I would like to give a few answers to this question, general ones, and some of them, the two first answers, are not specific to flies, but are more generally about how can we infer from different organisms to the human brain. So one general argument is about convergent evolution, which is the idea that we need to solve problems that flies also need to solve, and perhaps rodents also. For example, motion detection is something that virtually every organism, or almost all the organisms, probably need to solve. They need to solve it to find things that might be a risk for them or for other reasons. So it is possible that different organisms found the same solution to this basic problems, and by finding them in flies, we might then infer to humans. So this is one reason, and I think there are a few examples for where things like this happened, not just in the brain, but more generally in biology. I guess you heard some of these examples before. So this is one reason why it might work. But there is also the opposite view, which is that it is important to use a variety of modern organisms. People use mice a lot. It's the most widely spread model organism for learning the brain, because it shares a lot of the structure with humans, yet you can manipulate neurons and you can record from them. But mice are very different, or other rodents are very different from humans, and it's very well possible that in some cases they found local solutions, local in evolutionary timescales that are very different from what we found. And it is actually very interesting sometimes to find when different organisms solve the same problem in different ways. So if, for example, it is the case that flies and humans share some similarities in the neural structure, so in using specific chemicals in the synapses, but something is different in the way the inputs and outputs are distributed, then we should ask why is it? And it can teach us a lot about the brain. Why did our brain solve it in a different way? Is it accidental? Is it about the size of the brain? Is it about the complexity of the information that we have to process? So by comparing the solutions among different organisms, we can understand a lot about why our brain choose this versus the other solution, what could be an advantage of this. And there are, I think, a few nice examples for this. So it's interesting not only when it's exactly the same, but also when there are some differences, and it can help us to understand why these differences occur. So these are two things that are not just about flies. More specific to Drosophila, so Drosophila is used as a model organism in biology and to ask questions about genetics and development for around 140 years now, since Thomas Hunt Morgan started his fly lab. And people were skeptic already at the beginning, but I think it's clear now in genetic and development that some things that were found in flies turn out to be true for other organisms, including our own, including us. So some rules about genetics, some ideas about development were already found to be correct for other organisms. So I think in this, areas of biology, the answer is clear. And now people can say, okay, that's fine, but what about neuroscience? So, the same ideas that I told you before holds also for neuroscience, but more specifically, I think that we have more and more evidence from the last decades or more from the last years that there are actually some things that are similar. For example, you got a paper that refers a little bit to navigation. So the idea of heading cells that exist in rodents and also exist in flies and share some similarities. So we believe that it will turn out to be the case, that there are many similarities. But again, as I said before, even when we find differences, I think it can be very informative. So I think these are very general answers to this question. And as far as I'm concerned, we are done. If we have some small Q&As, I'll try to answer them, or we'll try to answer them later. But otherwise, does anyone else want to add anything?
No, thanks everyone for your questions. I think we've answered all of them. But if you happen to have others, or if you have questions about what we talked about today, or neuroscience, or our paths here, or anything else in general, feel free to email us. Our contact info is on the first slide that Lily showed, and it's been great having you. So thanks for your attention.